23 chromosomes. How many chromosomes does a cat have? 19. All right, 19 chromosomes for a cat. You get a prize. All right. Well, you guessed, didn't you? 19. So I think I'm going to rename this test to be. Oh, that's right. It was in there. I'm going to rename this test to be. You know, there's 23 and me. I'm going to rename it to be 19 and Mew. So we'll see. More thoughts. So. Yeah, that's the long story of the Burmese. Again, the Cat Health Network is still kind of there. All the chips got used up, so there's no more chips. So now we got to figure out, are we going to make more chips or sequence things? That's kind of where we are with cats right now.
In July. Cat Health Network meets in July.
Cat Health Network meets in July.
Sorry, early August.
Okay, early August, or what's going to happen. In the end, so do you know which laboratory was the most ready for these chips? Us. We got over half of those chips to do different projects. So we have done hypokalemia. That was the first one. From the time we got the chip, from the time we started prepping the DNA, which was last August, to October, we had the mutation. And Gandolfi found that one too, right? She's lovingly known as the Italian. So these things can need.
Explain that, because I didn't connect it from Wind Foundation.
That's the Italian. Yeah, that's the Italian right there. Yeah. We have localized 2 retinal degenerations, one in Bengal, one in Persian, and we're looking for the genes for those. We did the craniofacial defect, mediastinal lymphoma. We're doing the other brachiocephalic gene. Barbara's working on dominant white. Any dominant white cats? We need those guys for her project. We're doing things like polycystic kidney disease progression. So it's because of working with you guys over the past 20 years, our lab had the samples ready to use these chips. And so I think that's why we've had a pretty good year this past year.
If you look for HCM and Burmese, what do you need? Money?
HCM and Burmese, the number one thing we always need is the affected cats. That's always the hardest thing to come across, is the affected cats. We had Siberian breeders on, just yelling at us. We got part of it. And then when you start asking for those affected cats, suddenly they disappear into the woodwork.
Those
you know?
are his, not mine.
Yeah, exactly. They were his, not mine. That's his line, not my line, that type of thing. So once we get the samples, you know, the sample collection costs us nothing. It costs you guys. You have to do the echoes and things like that. It costs us nothing. Once we have the samples, that's when we have to talk about money, about putting somebody on the project. And the biggest cost, personnel time. Personnel time.
Do you know what percentage of the Burmese cats who actually tested? And when you're looking at the diversity, and we're at the wrong end of the scale, do you think it's possible that such a small percentage of Burmese cats were tested that they appeared more inbred than they actually are?
Well, no, because for other breeds we did about the same number of cats. So it was somewhere between 20 and 30 cats that we did. And so if that was true, then that would skew all the other ones as well, like for Korots or something like that. So as I was saying, we're pretty confident with 20 to 30 cats in a population, pretty much represents a population. Now we tried hard. We didn't take them all from one line, you know. We picked traditionals and contemporaries. You know, some of my cats are Tika cats as well. So if anything, it might be an overestimate of diversity and not an underestimate of diversity.
We have a great opportunity with Burmese, I think, because we have an unnatural split in that the traditional breeders haven't really been working with contemporary breeders, and they've been going up and down, you know, up generations. But now if we can come back together, it's probably not as good as going to Thailand, but
it's a step in the right direction. You know, you also have the break between the dilution guys and the non-dilution guys, you know. Yeah. So you have that break and then the US versus non-US. So you have a lot of fractionation, and that's probably why you're showing up that way, right? Is all that little fractionation and stuff. Yeah.
explain why search core results don't match up with the current test results?
No. Part of it was the interpretation of, do you call this a recessive trait or not? Okay, so you could play a game around that. Is this a recessive trait or not? Technically, it's a codominant trait, right? Because having one copy, you actually see the mutation, and then when you have two copies of it, you see a more extreme presentation. If you're just talking about when do I get craniofacial little monster kittens, takes two copies, then you would call it a recessive mutation. Search Core also called it incomplete dominant. I've never used the word incomplete dominant. I'm not sure what. Yeah, there's you could say. It's dominant with variable expression. You could say that as well. So when a cat has one copy, can you perfectly pick out that cat? No, because the facial structure is kind of variable and stuff. So that, I think, is the biggest differences with Search Core. And then I think with Search Core that they accidentally did use some contemporary cats in some of those crosses when they didn't know it, actually.
They never bred along those cats. They didn't try all the variations of the
So that all the different variations. And again, you know, it's just because you think you're picking a short-faced cat, that by just going by phenotype is not, you can't do it. You can't do it just by phenotype. So I think that's where it kind of went a little bit astray. But I don't think we're that far off. So I think it's an
piecemealed it out.
Well, I actually don't remember anymore. I haven't read the Search Core data in a long time. Yeah.
So what's the name of the gene that
I'm not going to tell you. Not until it's published.
Do you think that, okay, so it's the last 7 base pairs or 5 base pairs? Can you tell me if you think it's a no stop codon?
Whether it's a, what is the mutation?
12 base deletion in exon 2.
12 base pairs deleted in exon, yeah.
This is what I mean, oxy.
Yeah, so it's not a stop, but yeah. So I'm getting asked what is the gene and what is the exact mutation. I'm not going to tell you the gene, because then that means somebody else can develop the test, all right? All right, so that's what that means. And I know you wouldn't do that, but now it's actually out. Right. So now it's actually out on record. But then also because we want to develop a very good publication out of this as well. This is 20 years of work. We want a very cool publication out of this and stuff. And so we might be doing some other functional type of assays. So the mutation is 12 base pairs deleted out of exon 2. So exon 2, that means it's going to be very early in the protein. So it's taking 4 amino acids. So 12 base pairs, 1 amino acid is 3 base pairs. So it's taking 4 amino acids out of the protein, and that's probably really messing up the protein structure and leading to this defect. We would want, your favorite mutation is a stop mutation, where it breaks, it just stops the protein. But when you have this absolutely represented in a human, then you've, this is the gene, this is the right mutation, this is good and stuff. But so we're pretty confident about the mutation.
It's exon 2, so how many exons are there?
How many? 4. Okay. Yep.
Thank you.
Yep. And that's actually why they snuck behind my back. If it would have been 46 exons, like polycystic kidney disease, yes, 4 exons you can sneak, right?
Primers are not so noticeable.
Right, exactly. Yeah. So when you're ordering 4 sets of primers, I might not notice that. When you're ordering 46 pairs of primers, I'm like, where's that money come? Wait a minute, So so that's why you could sneak, right? Did you do a gene before it? And no. So hypokalemia, she did 2 genes. So the first gene was not right, and then moved to a second gene, and that was the right gene. But fortunately, both of those were small as well.
So-
No.
19.
19. Okay. Yeah.
So either the protein doesn't fold correctly, or it's lost a site where something
Right,
is-
these are the things we don't know yet.
Characterize that protein?
Right, these are the things we don't know yet, and that's what we'd like to see if we could figure out.
That would be very
Because, yeah, because the best published papers actually do something functional and actually figure out what the protein does. So we'll see whether we'll move in that direction or not. Yeah.
Yeah, I heard that you actually used Bombay to furnace.
Yes, one of these Bombays were really the secret to it all. Do you remember the cat's, well, we shouldn't say the cat's
Would
name.
that be because it was a non-carrier that?
No, he was a carrier, but he was a bit outcrossed. So remember I said there was this huge genetic region that we had, with 30 megabases or so, big genetic region. Well, because the Bombay was different on the ends, you could narrow it down, and so that helped Barbara say, okay, instead of looking through 30 megabases, which would have been hundreds of genes, really, what was your minimal area?
The Bombay reduces the region for 30 million bases to 700.
Yeah.
Wow.
Yeah.
Thank you.
Yep. Yep. Thank you, those Bombay breeders.
How did you prove to yourself that you had it once you thought you had it?
We had to test in a lot of affected cats. So one, you tested the pedigree. The parents have to be carriers. All the affected cats are always, all affected cats have to have 2 copies, right? They have to be homozygotes. That's what we have found to date, all cats. We have not found the mutation outside of American Burmese. And these are the standard things you do for anything, even like the glove mutation for Burmans. The Burmans have to be homozygote for it. You can't find the mutation in another breed. So we did you have to show it segregating in a pedigree. So we've gone through all that rigor to, and definitely is it not in the traditional guys and stuff? Is it not in Persians? Because is this just a gene that causes brachycephaly? No. So it's not the Persian gene, right? So we've had to go through all those different steps, and that's the data we have to generate and can continue to generate before we can publish the paper. And one of the things we'd like to do is get somewhat of an allele frequency in the population. Even though it'll be biased, it's interesting to show to people that this is a prominent mutation within the breed.
Because I wanted, when I sent my samples,
Mm-hmm.
and I had 2 contemporary non-carriers, and I wanted to make sure that they tested negative, because I wanted to make sure that you weren't just using traditionals for your non-carrier.
Right? Yep. No, we know to do stuff like that. Yep.
But I didn't know how you would identify contemporary non-carriers.
Right. Because I had, for example, samples from 20 years ago from Roger Horenstein. Right?
Yeah, those were traditionals.
Yeah, those were traditionals. Yeah. So,
So now I'm having nomenclature issues, because if you have a contemporary non-carrier,
Yes.
why is it still a?
Why it looks like a
Shouldn't we
contemporary. No,
be
not
saying
because it looks like a contemporary, because its
positive?
bloodlines are contemporary.
well, but she has a good point. Maybe we should stop saying the word traditional and contemporary. Well, isn't that a nice thought? All right. Woo! Let's start saying that. Here's another one that you might not like. Maybe we should stop saying NAB and UBC, FFC, whatever whatever thing, and make it Burmese. All right. So maybe we need to think about the little things that you can bust down the barriers with, right? So remember,
I'd rather call them carriers and non-carriers.
carriers and non-carriers. So. Because that defines everybody in the world. You're either a carrier or not, and it doesn't matter if you're a Tonkinese, an American, a Bureau, nobody, So, very good point to bring up, yeah. So, I think those are little things that maybe you should think about that might break down some barriers, yeah. But remember, I don't have a dog in this fight, right? I don't prefer contemporary over traditional whatsoever. I'm here to help make a breed healthier. And I think that's what you need to put on the board for every one of your meetings. What is your top goal? A healthier cat breed. And that